Hi, today I would like to discuss the results of my research project, the response of ants foraging activity and diversity to urban NDVI. Before delving into the topic, an overall concept needs to be stated that rapid urbanization in city regions has caused the degradations of ecosystems as a habitats, and which have affected the diversity of many plants and animals living in those areas because their original habitat has been barricaded by the buildings and roads. As you can see in the figure below, on the left, the compositions of Sydney urban area has over 60% of the total area in this region, which means that over 60% of those areas has become densely populated urban or suburban areas, causing the fragmentations of native plant covers with artifacts as you can see on the right. So, under the threat of urbanizations, several influences are exerted on important insect families, the ants. Firstly, the declined ant species and their abilities to participate in ecological functions such as involving food cycles as both predator and prey, spreading seeds of plants and clearing remains for both plants and animals, and secondly, effect on ants' ability to foraging and dominate the foods they find, and thirdly, such disturbance on the ecosystems would leave the gap in ecological niches for invasive ant species, as you can see on the as examples down below, native medians can be surrounded by a bunch of invasive Argentina ants. Based on the importance of ants' ecological functions and similar causes of plantation decline, the idea that factors of plantation cover can be used to indicate the diversity and forage activity of ants was inspired. And among the factors that indicate plantation covers, NDVI is relatively easy to use as it has many broad and convincing public databases. The objective of the experiments are to assess ant diversity and foraging behaviors in urban microsystems around the Sydney metropolitan areas from the following aspects: the total abundance and species richness, the Simpsons biodiversity, the foraging times, as well as the abundance and species richness of invasive species, and also to investigate the relationship of such variables to with local NDVIs. This study's hypothesis that higher regional NDVI would indicate higher total abundance, species richness, Simpsons diversities, and foraging times of the local ants, as the past studies shows that higher regional NDVI indicates more complex vegetation covers, which increase the available social niches and foraging times for the local ants, and also increase the complexities of habitats, causing lesser chance and longer times for them to search for certain foods. And at the same time, the increased diversities and activities of the native ants provide less available social niches for the introductions of those invasive species with alien identities, so their abundance and social richness will decrease relatively. The NDVI values was derived from the NDVI covariate data provided by SLGA database as it contains the long-term NDVI values from the atmosphere across Australia collected by satellites. And then the NDVI maps of Sydney regions will be drawn using the MapWheel package of R. Then 24 sampling sites were collected among these areas with 3 levels of NDVI, which were below 0.3, 0.3 to 0.6, and above 0.6. And sampling took place during March 12th to 28th, 2025, between the time periods of 3 p.m. to 7 p.m. with good weather without any rain or wind. Each sampling site has 3 sampling stations made with baking paper, containing baits, including sugar water, dissolved glutamine, and custard to attract ants, and the sampling will last for 1 hour, and all 6 kinds of data were collected. Then, the data collected were used as response variables and were summarized and visualized. In order to indicate the relationship significance between these response variables to the local NDVI as the predictor variables, GLM models in negative binomial, Gaussian, and Gaussian families were used. According to the results, 1,031 ants in 19 morphological species were founded. On the one hand, the most species were natives, as the blue part in the charts, but with relatively low abundance. On the other hand, 3 invasive species, as the orange part of the charts, were taking the majority of the total ant abundance, with a proportion of 54%. And according to a summary, most of the variables, including total abundance and species richness, foraging time, and invasive species abundance, have a low value among most of the sampling sites, and only few peaks value observed in others. Among GLM models between these variables and NDVI, there was no significant relationships between the most of variables with NDVI, except the abundance of invasive species that has a p-value less than 0.05. Other than that, an interesting observation is that outliers has altered the trend between the foraging times and NDVI. According to the result part, the ants observed have relatively low abundance, richness, and biodiversity in most of the sampling sites. This might contribute to ants' aggressive natures and their conflicts for food dominancy. You can see the photos up below showing they were fighting for food dominancy, and this might cause less abundance and richness on the station. Furthermore, the results show that ants usually spend less time finding food on the most of the sampling sites. This might be because that ants are relatively efficient in finding food using their pheromone communications, and the land cleaning among urban habitats is also reduced the barrier between ants and food bites. Also, invasive species which only occurs in few sampling sites or with huge abundance and relatively few species, not to mention the significant negative relationship between the abundance of invasive species and NDVI. The main reason of such a trend is that higher fertility and adaptability makes invasive species of ants more abundant than the local species in some sites, especially those with disturbed habitats. Just like this invasive species big-headed ants below that has multiple reproductive queens and in their nest allows them to have more workers, including their representative major workers, to fight for food dominance. And also, as mentioned above, the relationship between the forage time and NDVI was uncertain. This might indicate that other complicated factors would also influence the foraging time of ants, such as the impervious ground area, as you can see how impervious pavement roads is resisting meat ants to get through it without taking a risk of heat stress. Finally, there are some limitations and room of improvement of this study. The base stations were shown to be not very suitable for abundance and richness samplings, as the ants might escape from it. So involve the trap methods and perform diversity sampling separately with activity sampling may better meet the requirements and can also avoid sampling time period limits on the abundance and species richness of potential ants that can be observed. More replications of existing sampling may also give us a better vision for recent research. And the research results shows that NDVI isn't a good explainer for all variables, so further research direction can focus on more environmental factors caused by urbanizations, such as in previous space compositions or ground vegetation covers. At the same time, the diversity and behavioral factors of ants can be further extended, just like the relationship between the ratio of native and invasive ants could be considered, and the population changes in different species of ants during urbanization factors is also worthy of attention. And in terms of behavioral factors, the ability of ants to remove the food can also be considered.